I've been researching a protein called miraculin, which can change our perception of flavor. So one of the first things I did when a new protein atlas from the folks at BioHub came out was search for the miraculin protein and see if I could find any close matches in other plant species. Sure enough, there were a number of hits, including a few very close matches from plants that I could access. So I'll take you through the steps that I did to try and extract the protein from these plants and see if it had the same flavor modifying effects. Spoiler, it didn't, but here we go. Step one is to gather up some of the plant material and then chop it up and mix it with some water to hopefully extract out any of the extracellular protein into a liquid. So we use some water here with sodium chloride added to about 0.6 molar concentration to dissolve out these proteins. This is then filtered and then added to some cold ethanol. And what this does is it drastically lowers the solubility of the proteins, and so a bunch of them will crash out as a precipitate. We can then let this settle, collect it, and spin it down in a centrifuge to separate it from the ethanol. The ethanol and water and all of the dissolved salts and acids and things is poured off. More ethanol is added for a second wash. It's spun down again. The ethanol is once again poured off, and so hopefully we're left with a little pellet containing just the protein and a few other things that have also come out of solution. We can then add some water to this and put it on my tongue and then taste various sour things to see if it had an effect. Unfortunately, even all three of them, they didn't have any noticeable effect that I could see, and I ran a blind taste test between the extracted protein versus another set of extracted protein that had been heated, where hopefully if there was the case that one of those two was modifying the flavor, I'd be able to tell the difference. But unfortunately, it all tasted exactly the same as normal to me. So if there are these miraculin-like compounds in these plants, they're either in different parts of the plant than the ones that I extracted from, or they're in such low concentrations that they were indetectable, or most likely these similar proteins are similar in terms of structure. They have the same sort of trefoil shape, but they're not serving the same function. They have some different role, maybe anything from energy storage to some completely different metabolic function. So this was a fun experiment nonetheless, cool to see the protein extraction flow, but I'm a little bit sad that I don't have an easy alternative to miracle berries for accessing this cool protein. At least not yet. Stay tuned for a cool bioengineering project on that. Thanks for watching.